To the next lesson on molecular evidence for evolution. In this lesson, we're going to be looking at molecular homology and evidence for relatedness between species using DNA, amino acid sequences, mitochondrial DNA, the molecular clock, and DNA hybridization technique. So the number of techniques or molecular tools that can be used, or molecular homology can be used to determine relatedness between species are as follows, and we're going to be looking at those today. The first of which is chromosome banding pattern. So this is using chromosomes. They're stained with a certain stain and they reveal specific banding patterns. That's because the stain binds to certain regions of the chromosome which are rich in, say, G and C regions, guanine and cytosine regions. From there, they create different bands. Using these bands, we can see different species are related because they have similar staining patterns and similar banding patterns. This suggests they've all come from one common ancestor. There's also chromosome painting. That's where the chromosomes are coloured in a certain dye or colour, again, binding to different regions of the DNA in the chromosomes. And if you overlay the colour in those particular organisms, if they match up or are similar in the colour, that suggests that they are or were once related. There are other ways of looking at relatedness in species, and that's DNA sequences, protein sequences, and mitochondrial DNA. In this slide, we're going to be looking at DNA sequences and protein sequences. So, for example, a common ancestor has this particular nucleotide sequence. Over time, there are changes due to random mutations in various sequences, and this can be used to work out the relatedness between species. This common ancestor has only one difference to this species here, so therefore it's more similar to the common ancestor, whereas this species here has two differences in its DNA sequence compared to the common ancestor, so it's more distant. So the more time that goes on, the more mutations or the more types of mutations to occur, which will result in the greater difference. Protein sequences can also be used. So here is a particular example of a protein sequence from a histone H1, and these are the residues that it occurs. You can see between the human, the rat, the cow, and the chimp, there are various conserved sequences. So these sequences here are common between all types. That's due to them being highly functional or important for the structure of the protein. There are non-conserved sequences where they're different between different species, and semi-conserved. So between some species they're conserved but others are not. So using these, we can look at the differences between the species, and the more differences they have, the less related they'll be, or the more further back in time that they've diverged to form different species. Another technique we can use to work out molecular relatedness is called DNA hybridization. So in this process, we're looking at the genetic relatedness of the species based on DNA. So what's done in this process is two different separate species are used, and we have DNA of organism A and DNA of organism B. We heat those strands to break them apart. So when we heat them, it will break the hydrogen bonds between the two strands. When we break the hydrogen bonds, they'll form single strands. We'll then mix those two separate organisms together, and we'll allow or cool down the solution. This will enable hydrogen bonds to reform because the temperature enables it to occur, the hydrogen bonds to reform. Over time, those hydrogen bonds will reform. If there is complete hybridization, so every single hydrogen bond forms between each of those strands, we can say that those species are identical or related because they'll be having, or they'll share similar nucleotides which will bond to each other. So for example, if this nucleotide here was A and this one was T, they'd form a hydrogen bond together, which would create, and if every one of these was complementary, that would create a complete hybridization or an identical organism. If we only have partial hybridization, this suggests that the organisms are related because they share similar nucleotide sequences or share similar sequences in their genes. However, some of them are different because they have diverged or they're different species. If we have no hybridization, that means the organism is unrelated. So once the DNA has been heated and allowed to cool, it will form one of these three separate options here. The temperature at which these structures melt is used to determine the relatedness. So finally over here, we'll heat these particular options here, and the temperature at which they break apart is used to work out their relatedness or degree of relatedness. So for example, two separate species are mixed together, heated up, and we record the temperature at which they form single strands. So over here, we've got individual species A and individual species B, and we can see that individual or curve A has a higher degrees melting point. So the point at which the two strands will break apart. So there's one strand and there's another strand bound by hydrogen bonds. The point at which they break apart into single strands is known as the melting point here, Tm. As the melting point of curve A is higher, this suggests there are more hydrogen bonds between the two strands, which suggests that there are greater degree of relatedness between the species. Conversely, curve B has a lower melting point, which means there are less hydrogen bonds between the two strands. So less hydrogen bonds will be more like this or this here. Less hydrogen bonds, which means there'll be less energy required to break them apart. So you can see the melting temperature here is the point at which 50% of the DNA is single-stranded. So using this information, using the DNA sequences or the protein sequences, we can create these trees of relatedness. We can see the most common ancestor had these particular sequence, and we can see DNA mutations have occurred over time to show relatedness between species, which we'll cover more later. The next type of molecular homological technique, or the molecular technique we can use to work out how species are related, is known as mitochondrial DNA. So mitochondrial DNA is the DNA of mitochondria, no surprise. However, what's special about this is that mitochondrial DNA is only inherited from the mother, meaning it undergoes less recombination. Because if it goes from a mother down to all their children, this suggests, or this means, there'll be less recombination of that particular mitochondrial sequence, as it's not sharing the genes between a father. So chromosomal genes will mix and share different alleles between the mother and the father, or the parents. However, if mitochondrial DNA only is inherited from the mother, it has no recombination with the father. Therefore, the only recombination it's going to be having is mutations in that particular mitochondrial DNA. So using mitochondrial DNA, we can study various populations for specific types of sequences, DNA sequences that it has, and that can be used to look at the flow of genes or flow of populations over time. Take this picture over here. There are a number of groups known as a haplotype, which describe the type of alleles or type of DNA sequences in common groups of people. So here's the haplotype M, here's haplotype N, and IWX, and we know that this population or this mitochondrial DNA occurs in this geographical area that can be used to study evolution over time. Finally, the molecular clock is another molecular technique we can use. The molecular clock works on a prediction of the amino acids sequences in the changes they have over time. So for example, over time, amino acid substitutions will occur, and these substitutions occur at a steady rate. So here's a graph demonstrating the number of nucleotide substitutions that occur per millions of years. So as time increases, there'll be a greater number of nucleotide substitutions. As time decreases, there are less nucleotide substitutions. Based on this factor, we can predict the relatedness between species. So if there are a certain number of nucleotide substitutions between two species, so species A has, say, 15 or 20 differences to species B in terms of its relatedness, in terms of the nucleotide sequence. So you compare species B to species B, there's no differences, but species A to B, there's 15 differences in its nucleotide sequence. That can be used as a predictor to work out when roughly they diverged. The problem with using the molecular clock in this manner is that different proteins or different amino acids will tick or have mutations at different rates, and different proteins can differ in different groups of organisms. So it's a mechanism which can be used to create or look at differences between species based on the nucleotide substitution rate. However, that's not a rule that can apply to everything, so be cautious by using the molecular clock. This is the summary of what you need to know. And finally, this is the success criteria, so what you should be able to do at the end of this lesson. Thanks for watching.